Bismillahirrahmanirrahim and good morning. A very warm welcome and congratulations on your well-deserved success and also best wishes for your future. Let's talk about today's lecture. We will continue the chapter 3. In the last lecture we discussed properties of enzyme, structure of enzymes, model of enzymes, and energy activation. Today's chapter we will discuss factors affecting the rate of the enzymatic action. Then we will study about enzyme inhibition. Next we will discuss the classification of enzymes, and then we will discuss exercise, and in the last there is homework. Now the first thing is factor affecting the rate of the enzyme action. There are certain external conditions which affect the rate of the enzyme reaction. These are temperature, pH, enzyme concentration, substrate concentration. Before moving on the topic, there are certain important points, cardinal points. The first one is minimum, the second is optimum, and the last is maximum. Now, what is minimum? Minimum is the condition which is least or in a very small amount. And when we talk about maximum, in maximum it shows greatest amount. And when we talk about optimum basically it covers most favorable situation. Now we will discuss temperature. What is temperature? Basically the temperature is the average kinetic energy of the particles in a system. So, on heating, there is increase in molecular motion of that system. Molecules of substrate and enzymes move more quickly so that the probability of reaction to occur is increased. Increasing the temperature up to 10 degrees Celsius in existing temperature doubles the rate of the reaction. Means when temperature is increasing up to 10 degrees centigrade in existing temperature, it doubles the rate of the reaction. But this effect is up to certain limit. Now in this image you can see that now it's cold condition, but when it gets hot there is continuous and there is random motion in the molecules. Temperature that promotes the maximum activity is called optimum temperature. As we have already learned that optimum temperature is the most favorable condition, the most favorable temperature. Increase in temperature above this level, which level means this optimum level, decreases the rate of reaction. ऑप्टिमम टेम्परेचर पे पहुंचने के बाद जब मज़ीद इंक्रीज होता है टेम्परेचर तो क्या होगा, रेट ऑफ रिएक्शन डिक्रीज हो जाएगा. नाउ व्हाट इज़ द रेट ऑफ रिएक्शन? द रिएक्शन रेट और द रेट ऑफ रिएक्शन इज़ द स्पीड which a chemical reaction take place. Due to change in secondary and tertiary structure of enzyme are disturbed. Means when temperature is increased, the protein structure which is secondary and tertiary structure of enzymes are disturbed. Now what is secondary and tertiary structure? Basically these secondary and tertiary structure are the structural conformation of protein, or you can say these are the pattern of formation of protein. And at that time enzyme is said to be denatured. उस टाइम पे enzyme denatured हो जाएगा, means it take away or alter the natural qualities of that specific enzyme. Enzymes unfold and structure of active site is lost. In the figure you can see that this is native state and here is the active site. But when the temperature rises, the enzymes become denatured, and in the denatured situation the enzymes unfold and the structure of the active site is lost. Temperature and enzyme activity. Temperature cause the denaturation of enzyme is called maximum temperature. That temperature which basically cause the denaturation of enzymes, which basically take away the property of enzyme, is known as maximum temperature. Maximum enzymes are most active at optimum temperature, and in human being usually it is 37 degrees Celsius. A few bacteria have enzymes that can withdraw very high temperature up to 70 degrees Celsius. Such enzymes are used, means which enzymes which are working on this 70 degrees Celsius. Such enzymes are used in biological washing powders for high temperature washing. Now, what is biological washing powder? Biological washing powder and liquid contain enzymes and proteins to get clothes clean. Here are some properties of washing powder. Basically, they contain proteases and lipase, work best at low temperature, that is 40 degrees centigrade. Here we are talking about the certain enzymes which are working on high temperature. So we are mentioning here that these are used in high temperature washes, more effective than ordinary detergent. Now here is a graph which basically show the activity of enzyme on certain temperature. On x-axis temperature is shown, that is 0, 10 degrees Celsius, 20, 30, 40, 50 and 60. And here is the rate of reaction. As we have defined what is the rate of reaction, basically it is the speed at which a chemical reaction is taking place. Now when the reaction is start and as the temperature is moving, so the heat energy causes more collision between enzymes and substrate. Then at optimum temperature of human is close to 37 degrees centigrade. Now you can see that here the graph is shown at its peak, which is optimum temperature. But after availing the optimum temperature, when there is certain increase in temperature, it makes the enzymes denature. As it is mentioned here, enzymes denatured at high temperature, so the rate of the reaction falls rapidly. As you can see that here the graph is showing that the rate falls rapidly. Now the next is pH. What is basically pH? pH is basically the concentration of hydrogen ion. Every enzyme has particular pH range. The range at which maximum rate of the reaction is achieved is called optimum pH. बिल्कुल स्टार्ट में हमने कहा था there are some important points: maximum, minimum and optimum. So here the range at which maximum rate of reaction is achieved, it is called as optimum pH. The change of pH will lead to ionization of amino acid. As you know that enzymes are made up of protein, and proteins, amino acids जो हैं वो क्या करते हैं? They help in the formation of protein. Now there is ionization of amino acid atom and molecules change the shape and structure of protein. so damaging the function of protein. By increasing the pH, basically वहाँ पे क्या हो जाएगा, जब pH increase होगा तो it damage the shape and structure of protein. So after damaging the shape and structure of protein, they become inactive temporarily. Now you can see that there is hydrogen ion, means hydrogen ion concentration is getting increased. Here is optimal pH. In optimal pH, the substrate combine and fit here at this active site. But as the pH of the enzyme is optimum, the shape of the active site gets changed, and so that this substrate can't get entry. At the pH from the enzyme optimum, the shape of the active site changes so severely that enzymes cannot work. Now the pH. Optimum pH value for the most of the enzymes falls in the range of pH 6 to 8. Some enzyme like papain from green papaya act both in acidic and basic. Now this is the important point. You can underline it on your book. Digesting enzymes like pepsin is active at pH 2 and trypsin show maximum activity at pH 8. Now here is the graph. At the x-axis the pH is shown, that is 4, 5, 6, 7, 8, and we have talked that the maximum range or the optimum range for most of the enzyme is between 6 to 8. You can see that at this condition, means pH 6 and pH 8, there is a peak point, there is the optimum rate of reaction, but at the pH 4, 5, it just starts, it is low, and after taking this peak, after getting this optimum range, then the pH again falls, because this pH means the greater hydrogen ion concentration basically changes the position, changes the shape of the proteins. Now the next point is enzyme concentration and reaction rate. How the enzyme concentration affect the reaction rate. The rate of the reaction increases as enzyme concentration increases. Reaction rate increase होता जाएगा जैसे हमारे पास enzyme की concentration ज़्यादा होगी. What is the reason? Because उसको maximum active sites मिलेंगी, और जब maximum active sites मिलेंगी तो क्या होगा कि reaction rate ज़्यादा होगा. But इसके लिए क्या होगा कि pH and temperature are constant. At higher enzyme concentration, more enzymes are available to catalyze the reaction as more active sites are available. More reaction at once. There is linear relationship between reaction rate and the enzyme concentration. When we draw the graph, we will get linear relationship. It has a direct relationship. Why? Because as the enzyme concentration increases, the rate of the reaction increases. Now you can see that in this graph, as the enzyme concentration increases, the rate of the reaction is also increases. Now the next point is substrate concentration. Increasing substrate concentration increases the rate of the reaction. But again, pH, temperature are constant and the enzyme concentration is maintained at high level. The increase in substrate concentration, which is shown by S, increases the velocity of the enzymatic reaction at first. When the substrate increases, it will increase the velocity of enzymatic reaction. The reaction ultimately reaches a maximum velocity at equilibrium state. So the rise in V means velocity is decreased with further increase in S. As the substrate concentration increasing as it gets the optimum level. After getting the optimum level, when the reaction reaches at maximum velocity at equilibrium, फिर क्या होगा? There rise in V. When the velocity is increased, there is decreased and further increase in substrate. Reaction does not increase by further rise in substrate concentration. Now what is the reason? Because all active sites are occupied by substrate and no enzyme is left free to bind with additional molecules. Now here is the graph for substrate concentration. Here is substrate concentration and here is initial rate of reaction. So increased substrate concentration leads to increased rate of reaction. When the substrate concentration is increased, it will cause the reaction rate at maximum level. But when at optimum level, after getting the optimum level, and when there is certain increase in substrate, then it will affect the rate of reaction. As all enzymes are present, are working at maximum rate. जब सब enzymes वहाँ पे काम कर रहे होंगे तो substrate की amount को increase करने का as such कोई फ़ायदा नहीं होगा. Now the next topic is enzyme inhibition. Now what is enzyme inhibition? इससे पहले हम लोगों ने enzyme की activity पे बात की. अब here is inhibition, means there is something to inhibit the reaction. Now what are these? The phenomena in which an enzymes fail to catalyze a reaction is called enzyme inhibition. Okay, this is the phenomena in which enzymes fail to work out, fail to produce the product. This is known as enzyme inhibition. Sorry, I have to discuss this figure. Now there is a flow sheet diagram. The first is shown that here is enzyme inhibitors. Now there are two types of enzyme inhibitor. The first one is specific and the other one is non-specific reason. Now what are specific? In the specific there are irreversible, reversible, non-competitive, allostatic and competitive. We are discussing all these shortly. But first we talk about the non-specific. What is the non-specific one? The non-specific are basically, or you can say that the non-specific method of inhibition includes any physical or chemical change which basically denature the protein, which denature the activities of protein. It may be include acid or base, means when the pH is changed, when the temperature is changed, when the concentration of alcohol or heavy metal or reducing agent, when all these factors are affecting, it may act as non-specific enzyme inhibitors. Now we will talk about specific enzyme inhibitors one by one. Enzyme inhibitors. What are enzyme inhibitors? The molecules which react with enzymes but are not converted into desired products are called enzyme inhibitors. Those elements which are inhibiting the enzymes to complete their reaction, these are known as enzyme inhibitors. For example, poison, cyanides, antibiotics, antimetabolites, penicillin and sulfur drugs. Now you can see that here is enzyme. The red color is showing basically that this is the substrate. Means substrate comes and get entry here this active site. But sometimes it happens that there are certain elements which are known as inhibitors. They get attached to another point which is known as allosteric site. and they inhibit the formation of enzymes product complex, or you can say that the conversion of substrate into product. Now we will talk about all these enzyme inhibitors one by one. The first one is poison. As all of you know that what is poison? Poison are the substance that causes the death. Now the next is cyanide. What are cyanides? Cyanide is the chemical compound that contains the group carbon with triple bond nitrogen. It prevents the cell of the body from using oxygen. Basically, it is an irreversible enzyme inhibitor. It prevents the cytochrome oxidase from doing its job, which is to transport electrons in electron transport chain. Without the ability to use oxygen, mitochondria cannot produce energy carrier, that is ATP. Tissues that require this form of energy, for example heart, muscle cells, nerve cells, quickly expend their all energy and start to die. So it is also act as enzyme inhibitor. Now antibody. Antibody is a protein component of immune system that circulates in blood, recognizes the foreign substance like bacteria and viruses. Next is sulfur drug. Sulfur drug are called sulfonamides. Any member of the group of synthetic antibiotics containing the sulfonamide molecular structure. Now the competitive inhibition, a type in which enzyme activity is blocked by the presence of a chemical that compete with substrate for binding to the active site, is called active inhibition. Means enzyme activity is blocked. How it is blocked? By the presence of chemical that compete with substrate for binding the active site, is called competitive inhibition. Structurally it is normal to the substrate. ये दिखता बिल्कुल वैसे ही है, ठीक है, ये वैसे ही होता है substrate जैसा, and fits into the active site of the enzymes, but not similar enough to substitute fully for normal substrate in the chemical reaction. It looks like the substrate, but it not substitute the substrate in the chemical reaction, and enzymes cannot it to form the reaction product. Now it is temporary and inhibitor eventually leaves the enzyme, so it is called reversible inhibition. Means as soon as the conditions are favorable, जैसे ही वो inhibitor हटेगा तो दोबारा से क्या है, enzyme अपना काम start कर देगा. That is the reason it is called as reversible inhibition. This means that the level of inhibition depends on relative concentration of substrate and inhibitor. Here is a figure in which the enzyme is shown and here is substrate. Now the competitive inhibitor. We are saying that this inhibitor looks like the substrate, not exact one. It cannot substitute the substrate. When it combines here, and it will prevent the active site to get entry at the active site. As it prevents, so the product is not released. Enzyme substrate complex is not formed. Enzyme product complex is not formed, and product is not released. Next is reversible competitive inhibitor. We are discussing it shortly. What is reversible competitive inhibitor? At this, now this is the enzyme and this is substrate. When the reversible competitive inhibitor come and get entry at this active site, increase in substrate concentration. Now it may happen that this you can say that inhibitors when it leaves the enzymes, then there is maximum amount of substrate is present there, and that substrate get entry at its active site, at its specific active site. Now here is the example of competitive inhibition. In this competitive inhibition, here is malonate. This is the enzyme, and at this enzyme the substrate is succinate. When this succinate combines and get entry at here at the active site, it will form succinate enzyme complex, and as the product fumarate is released with enzymes. But sometimes it happens that malonate comes and get entry with this enzyme instead of succinate. If you see the structure, that C double bond with O, single bond with O, then CH2, CH2, then there is C double bonded with O and single bonded with O. There is a little bit difference, and that difference is here, that CH, then this CH2 is missing. Now it is all, some extent it is similar to, this malonate is similar to succinate, but it cannot substitute the succinate. So when this malonate come and combine this enzyme, it will form malonate enzyme complex. But as this is not the exact one, this is not the exact reactant, so there is no product is formed. An example of inhibitor is malonate. Succinate dehydrogenase catalyzes the formation of fumarate from succinate is completely inhibited by malonate. The enzyme which is used in this reaction is succinate dehydrogenase. Now, what is the importance of competitive inhibitors? The first is that it supports lock and key model, as you have already studied it, that for every specific lock there is specific key. It shows that substances which are similar to the substrate are not acted upon the enzymes. ये ज़रूरी नहीं है कि अगर वो उन जैसे हैं तो वो फंक्शन, वो वाला प्रोडक्ट भी प्रोड्यूस करें, ठीक है. They can't substitute the specific substrate. Used as drugs in order to control the pathogens. Now, what are pathogens? Which depend upon living things for their condition. They cause disease. Now, the next is sulfonamides are used to fight bacterial infection. These sulfonamides are used for treating urinary tract infection, bronchitis, or eye infection. Now here are different uses of sulfonamides. They can be used as antibiotic agents, antiretrovirals, diuretic, dermatological. It can be used as hepatitis C antiviral, children antibacterial drugs. Now the next is non-competitive inhibitors. First we discussed the competitive inhibitors. Now we are discussing non-competitive. And in competitive there were two types. One is reversible and other one is non-reversible. Now in non-competitive inhibitors, inhibitor molecules bind to the enzymes other than active site. आपने देखा होगा कि competitive inhibitors में क्या करता है कि वो active site को cover कर लेता है. But in this it will bind other position. The other binding site is called allosteric site. They inactivate the enzyme temporarily, means reversible inhibition, or denature the enzymes permanently. Here again there are two types. One is reversible and other one is irreversible. If the enzymes get the favorable condition and if the inhibitors leave the enzyme, at that time it will become reversible inhibition, means वो temporarily basis पे था. And if it gets entry permanently, then it denature the enzyme. Now the reversible non-competitive enzyme work not by permitting the preventing the formation of enzyme substrate complex, but by preventing the formation of enzyme product complexes. You can see here that this is the enzyme and this is the active site. Now it may happen that substrate get entry here. It may attach to the enzymes, but sometimes there are certain inhibitors which come and get entry to the other side, which is known as allosteric site. Now irreversible non-competitive inhibitors. They destroy enzyme by altering its shape. For example, cyanides and salts of heavy metals. Now, cyanide causes the death of organism by inhibiting cytochrome oxidase, essential for cellular respiration, as I have already discussed it. They block the action of enzyme by combining with iron, which may prevent in the prostatic group. You have studied about cofactors. कोफैक्टर में आपने प्रोस्टैटिक ग्रुप पढ़ा था और उसमें प्रोस्टैटिक ग्रुप में क्या है मीन्स मे बी इट कॉन्टेन आयरन एंड देन दिस आयरन बेसिकली ब्लॉक द एक्शन ऑफ एंजाइम. Irreversible non-competitive inhibitors. The second type is salt of heavy metals. Now the salt of heavy metals such as mercury, silver and copper combine with thiol group in enzyme, breaking the disulfide bridge. Basically what it does, it breaks the disulfide bridge. Self-hydral group, SH is basically that is self-hydral group of protein. For example arsenic, अगर हम इसके example में देखें, as these bridges are broken, enzymes become denatured and inactive. Now this is self-hydral group, means this is SH. Here is the definition, that functional group consisting of an atom of sulfur bound into an atom of hydrogen. Helps stabilize structure of protein. Basic its function is that it gives stabilization to the structure of protein. Disulfide bridges in tertiary structure of protein, called thiols when found in organic compounds. Now the next topic is feedback inhibition. What is the next topic? Basically it is feedback inhibition. Now what is feedback inhibition? Feedback inhibition is basically the activity of almost every enzyme in a cell can be regulated by its product. When the activity of enzyme is inhibited by its own product, ठीक है, it is known as feedback inhibition. जब हम कहते हैं कि हमें फीडबैक दें, जब हम आपको कुछ चीज़ समझाते हैं और उसके बाद हमें कहते हैं कि हमें फीडबैक मिलना चाहिए, कैसा है, मीन्स व्हाट इज़ द प्रोडक्ट, आपको उसकी समझ आई या नहीं आई, आपको ऑब्जेक्टिव्स क्लियर हुए हैं या नहीं हुए, ठीक है ना. तो ये जो फीडबैक बेसिकली है, मीन्स वो जो प्रोडक्ट होगा, वही प्रोडक्ट जब इनहिबिट करता है एंजाइम एक्टिविटी को, इट इज़ नोन एज फीडबैक इनहिबिशन. नाउ वी विल रीड फ्रॉम स्लाइड. Inhibition of the activity of enzyme of a biosynthetic pathway by the end product means this passes through a biosynthetic pathway. Different actions उसके अंदर होंगे, ठीक है. By the end product of that pathway, now this end product, it will inhibit the action of enzyme activity, is called as feedback inhibition. For example, formation of substance D. Now in this equation you can see that here is substance A, B, C, D. Now D is product. Substance D from A is catalyzed by 3 enzymes, that is E1. E1 जब act हुआ तो it will convert it into B. Then B is converted into C with the help of E2. And then C is converted into D with the help of E3. When enough D is formed, it inhibits the activity of E1. When the concentration of D is getting high, then this D will inhibit the action of this A1. Inhibiting the E1 deregulates its own synthesis. Here is the example from your book. In this example, here is aspirate, which is molecule A, and here is the final product, which is known as threonine. Now, when the threonine means the end product of this pathway is present in excess, when it is present in excess amount, it binds to the allosteric sites. It binds to the allosteric site of aspirate. When all the threonine is consumed in cellular events, the threonine molecules which is attached to the allosteric site is also removed. The pathway resumes its activity once again. Now here is the video.
Enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction. If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
So you can see that here is the product, और आप ये देख सकते हो कि उसका number increase हो चुका था, ठीक है? Then these products come and attach to the another site than active site, which is allosteric site. Now the next topic is classification of enzymes. An enzyme can be classified. Now there are basic two criteria. The first one is basis of reaction type, and the second one is on the basis of substrate. This and with the suffix that is ase. Now first we will discuss on the basis of classification on the basis of reaction type. It is given by International Union of Biochemistry in 1961. Now this one is important for MCQs. The first one is oxidoreductases. As the name shows, there is oxidation reduction process. These are involved in redox reaction. They catalyze the transfer of hydrogen or oxygen atom or electrons from one substrate to another, also called as oxidases, dehydrogenases or reductases. For example, dehydrogenases, that is the removal of hydrogen, and oxidase means add oxygen to hydrogen forming water. Now this is the journal formula. In this, here is molecule A, reduction plus B oxidation, and then it will give A is oxidized and B is reduced. Now the next is second one is transferases. Now what are transferases? Basically it catalyzes the group transfer reaction. They transfer a group of atom from one molecule to another. For example, transaminases. It transfer amino group from one molecule to another, and there is the another group that is phosphotransferases, transfer of phosphate group. Now you can see in this example that this is glucose molecule, and here it is the structure formula. You can see that there is OH bond, but when this reaction is going on, hexokinase help it out. ATP, you can see that this ATP means adenosine triphosphate is converted into ADP, means one phosphate is losing here. Now this phosphate come and combine here, okay, and it form glucose 6-phosphate. Now the next is hydrolases. Catalyze the hydrolysis of substrate by addition of water. Journal equation is A, B or X, यहाँ पे कुछ भी हो सकता है, means there are two reactants and there is addition of water, and then you can say in the product H and X, then OH plus HA is formed. ठीक है, these reactions are reversible. For example, maltase broken to glucose, and lipase, lipids are broken to fatty acid and glycerol. Now the next one is lysis. As the name show, means there is something which is breaking, so breaks the chemical bond without hydrolysis. General equation is AB is converted into product A plus B. For example, decarboxylase means remove carboxyl group from the respiratory substrate to release carbon dioxide. There is no release of water molecule. Here is fumarate. Fumarate is converted into L-malate with the help of fumarase. Now the next is isomerase. Isomerase, these enzymes, so यहाँ पे ये Z जो है that is missing. These enzymes bring about intracellular rearrangement of atoms in the molecules and forming one isomer from another. That is A, B and C. These are reactant. These reactants are converted into A, C, B. Basically there is a rearrangement of atoms. As you can see that here is glucose, as you can see that here is glucose 6-phosphate, and here is a structure formula now, and it is fructose 6-phosphate. Now there is a little bit difference in their structure. There is a rearrangement of atoms. Now the next is ligases. Basically, it catalyzes the synthesis of new chemical bonds using ATP. For example, DNA ligase is involved in DNA synthesis. Here is the journal formula. And what is the journal formula? That reactant A, B using the ATP, and it converts into product A and B, and ATP is converted into ADP with the release of inorganic phosphate. Now here is the table. Oxidoreductases reaction catalyzed. किस तरह के reaction को catalyze करते हैं? Transfer of hydrogen and oxygen atom or electron from one substrate to another. Example is dehydrogenases or oxidases. Next, transferases. Transfer of specific group of phosphate or methyl from one substrate to another. And its example is transaminase. Then hydrolysis of substrate, esterase or digestive enzyme, then isomerase, change of molecular form of substrate. Phosphohexoisomerase or fumarase is the example. Then fifth one is lysis. Now what is lysis? Non-hydrolytic removal of group or addition of a group to the substrate. Decarboxylases, aldolases. And the last is ligases, also known as synthetases. Joining of two molecules by the formation of new bond, for example, citric acid synthetase. Now, there is another classification which is based on substrate. Enzymes can be classified on the basis of substrate they use. Some of the examples are proteases, lipases, carbohydrases and nucleases. The first one is carbohydrases. Now what is carbohydrases? These enzymes can break down of these enzymes cause the breakdown of carbohydrates. There are certain examples of carbohydrases, for example amylases which digest the starch or glycogen into maltose. Then there is cellulase, digest cellulose, maltase which digests maltose into glucose. Then sucrase, we digest the sucrose, and lactase, which digest the lactose into galactose or glucose. The next one is protein. Now the enzymes can act upon protein, and these are known as proteinases. Examples are pepsin or trypsin or carboxypeptidases. Next is lipids, and the enzymes are known as lipases. These enzymes hydrolyze lipids into fatty acid and glycerol, for example pancreatic lipase. The next is nucleases, means the substrate is nucleic acid. Now the nucleases, these are involved in the breakdown of DNA and RNA, as the name shows. ठीक है ना? Examples are RNases digest RNA in ribonucleotides, DNase digest the DNA, ATPases, basically it causes hydrolysis of ATP in muscle. Then there are certain example, that is urea, and the enzyme is known as urease. Sucrose, it is known as sucrase. Now girls, our chapter is completed, and here is exercise. The first question is MCQs. Here is the answers of all MCQs. You can note down it, and then there is a homework that you have to do short questions from 2 to 21. I will share the page number in the WhatsApp group, and you will do it on your neatly on your registers. Thank you girls, thank you so much. Take care.
During the normal enzyme catalytic cycle, the substrate encounters an enzyme with a specific active site to which it binds, forming an enzyme-substrate complex. The enzyme then facilitates the breakdown of the substrate to its products, which part from the enzyme, leaving the active site free to catalyze another substrate as the cycle begins again. Competitive inhibition occurs when an enzyme encounters a blocker, which mimics the properties of the substrate and binds to the enzyme's active site. Thus, when the substrate is encountered, the active site is not available for attachment and no reaction will occur. Non-competitive enzyme inhibition involves the binding of a blocker to the enzyme away from the active site. This binding causes a conformational change in the enzyme, altering the shape of the active site, which prevents the substrate from binding. No reaction will occur as long as the non-competitive blocker is bound to the enzyme.